starting, and on the off chance that you are not a neuroscientist, neurons are the cells that make up your central nervous system. Neurons are not the only cells we have in our brain. We also have neuroglial cells, which do not participate directly in synaptic interactions and electrical signaling, although their supportive function helps define synaptic contact and maintain the signaling abilities of a neuron. Glia are actually more numerous than nerve cells in the brain. Glia roles that are well established include maintaining the ionic milieu of nerve cells, modulating the rate of nerve signal propagation, modulating synaptic action by controlling the uptake of neurotransmitters, providing a scaffold for some aspects of neural development, and aiding in or preventing, in some instances, recovery from neural injury. There are 3 types of glial cells in the mature central nervous system: astrocytes, oligodendrocytes, and microglia. Astrocytes are the main class of glial cells in the central nervous system. During development, astrocytes secrete multiple signals that induce formation or elimination of synapses and regulate synaptic strength. It is important to mention that this is not the only function they have. However, here we will emphasize on it due to its importance in brain plasticity. So you may be wondering, what are synapses? Synapses are tiny connections between the neurons in your brain that, when they are functioning properly, allow neurons to communicate with each other. Each neuron has multiple synapses connecting to multiple other neurons. As a result, we have literally trillions of synapses. An excitatory synapse is a synapse in which the message and action potential in a presynaptic neuron increases the probability of an action potential occurring in a postsynaptic cell or neuron. So, when we're born, our brains have a great deal of flexibility. Having this flexibility to grow and change gives the immature brain the ability to adapt to new experiences and organize its interconnecting web of neural circuits. As we age, this quality, called plasticity, decreases. Nature has evolved these circuits to become more stable and less flexible. Otherwise, our brains would not mature and we would experience our whole life like a young child does. In the developing brain, immature synapses contain calcium-permeable AMPA glutamate receptors, which conduct fast synaptic signals at excitatory glutamatergic synapses. These are excitatory synapses that use glutamate as a neurotransmitter. These receptors are subsequently replaced with GluA2 containing calcium impermeable AMPA receptors as synapses stabilize and mature. This switch in AMPA receptors contribute to synapse maturation and stabilization by decreasing the entry of calcium into the postsynaptic cells. This is important because it limits the plasticity mechanism that requires signaling through calcium-dependent pathways. This essential switch in the AMPA receptors and neural synapse maturation is regulated by astrocytes, which secrete chording-like 1. So, to make it more clear, this is like the entrance to a disco in which the person at the door is very permissive and lets calcium enter very easily. As time passes, the manager decides to change the person in the door to a more strict person, and the entry of calcium is decreased. How do neurons know to switch the calcium permeable to calcium impermeable receptors? Well, the manager, which are astrocytes, notifies the person at the door by giving it chording-like 1. This new role of astrocytes was not previously known. It was known that astrocytes are important for development of the brain. However, very little was known about the role of astrocytes in the adult brain. It's important to study brain plasticity because it teaches us how the brain remodels itself in response to new experiences. These findings could point the way to restore connections that have been lost due to aging or trauma.